I'm in the mycology lab where I have been doing a lot of work on taxonomy, diversity, ecology and biology of Termitomyces species of Goa. This is 7 January, Friday 2011. The new year has just begun and the new year has brought with it once again a gift of Termitomyces mushrooms, that too from my own garden near my house at Santa Cruz, Bondir, Tiswadi Taluka in North Goa district in India. What is interesting about this, what the specimen which we have collected, there were some 80-90 specimens of mushrooms, Termitomyces mushrooms. Termitomyces mushrooms never come in Goa during the month of January. This is for the first time that I have recorded this Termitomyces petaloides fruiting in January. Earlier in December and January, so two continuous years, in 2008 and 9 I have collected specimens of Termitomyces from the same habitat in the same garden, and that has opened absolutely a new area in terms of the impact of climate change in Termitomyces, and specifically whether Termitomyces speciation or new species formation is regulated by some climatic factors, and especially whether there are climatically well adapted and adjusted species like this species of Termitomyces petaloides, on which we are working before the publication of the new taxa. This has got a very interesting type of petaloid or split type of margin with a very prominent umbo, fully melanized. It rolls up. It's very hard because it comes through thick layer of sand. The whole strata is sandy, and we have named it petaloides because of its particular interesting type of these epicuties you can see, and petaloid, split petal like, flower petal like margin you can see, and this is the central umbo, and it is pale pinkish on maturity but creamish when it is white. It has got a superior annulus on the top, which shows it is a membranous, remains of membranous well, and is about a thick tapering stripe also. But most interesting is I have uprooted this along with the pseudorhiza. You can see here there is a small basal disc. This basal disc is a connection of the entire fruit body to the fungus comb, underground fungus comb. So from this point the nutrients are translocated. This is the hypogeal part which is hollow. You can see this hollowness. It is like a empty rubber tube. It has melanized here. This is deposition of fungal melanin in these spots wherever it has encountered some obstacle or wherever it has grazed against some pebble or some sort of hard surface. Then once it touches the soil, then it becomes solid, and then it forms the solid stipe portion. So this is the stipe portion. This is epigeal part of the specimen. This is a hypogeal pseudorhizal part of the specimen. Now this part, hypogeal part, supports the pileus before it matures and then discharges the basidiospores from the gills. So other characteristics are known. Now, commenting on the relationship between climate change and evolution of new species in the state of Goa, we have now definite evidence that in January this species is fruiting. That means the phenology or fruiting periodicity of Termitomyces petaloides is governed by winter conditions. Whereas these species start fruiting in Goa, other species, there are some 25 to 30 species which I have recorded in Goa, out of which 5 to 7 taxa are new to science, and there are several new reports for Goa and India. What I have found consistently is that Termitomyces petaloides has evolved to fruit under low temperature conditions. That is, the maximum temperature during January is about 27 to 29 degrees Celsius. The minimum temperature ranges from anywhere between 14 degrees to 22 degrees. So this is actually not favorable to fruiting of the Termitomyces species, which generally fruit when the temperature, daily maximum temperature is above 30 degrees consistently, and the minimum temperature falls to anywhere between 22 degrees to 28 degrees between the month of July to September. The peak period of Termitomyces fruiting recorded for so many years and traditionally known also is July and August, when definitely we do not have winter-like conditions. Besides, these are months of very high humidity, almost 85 to 99 to 100%, and the humidity during the winter season, that is in January today, the percentage relative humidity is hardly 55 to 65%. With such moderate to low humidity conditions, it is really amazing that the Termitomyces genus, the whole Termitomyces genome, is interacting with the climate change in such a dynamic manner that in this part of Goa, Tiswadi Taluka, in soft sandy substratum, the subterranean fungus forms of Odontotermis and Macrotermis, most possibly these are the two termite taxa, which have created small fungal gardens underground, and they are producing these fruit bodies, epigeny, during the month of January, and thereby putting up a challenge to mycologists, ecologists as well as climate scientists, how these macrofungi, especially the mutualistic macrofungi, are adjusting, adapting and responding to the climate change. In the sense, the phenological spectrum has shifted, and it has been known it has been shifted in angiosperms or flowering plants because of climate change. But here we have a case where fruiting of generally species adapted to high moisture condition, high soil moisture condition, high aeration and higher temperatures, that is between 25 to 35 degrees, which occurs between July and August, then suddenly the same genus produces a species which is well adapted to winter-like condition. So the question at the end of this whole exercise, after studying the pattern of flowering, after studying the emergence of this number of species in the natural habitat, and after examining this biologically and taxonomically, the final conclusion is that genes may respond to environmental challenges in such a manner that mutualistic species would be able to respond by speciation, because it has been known that from the same fungus comb it is possible to get more than one type of species. So here is a concrete example for the first time in the world of speciation triggered by climate change and being sustained for not one but continuously for 3 years. Thank you very much.